grade 8. Number 1, grade 7 learners were asked to name some basic science skills. Give 5 examples of skills they are likely to name. Now, when you look at topic 1 of integrated science grade 7, there is a substrand called the skills, the basic science what? Basic science skills. Now, we are giving the examples. Number 1, we have observation skills. Observation skills. Number 2, manipulative skills. Manipulative. Always check the spellings. That is another key area that you as a learner, you need to put more effort on. Make sure that the spellings are correct. Number 2, we have measuring skills or measurement. Measuring skills. Number 4, we have conclusion skills. Number last. Now we have a lot of skills. We have data analysis. That is also a skill. We have observation, manipulative, measuring skills, conclusion skills, data analysis. Those are all skills. Experimentation. Those are still skills. Now, those skills are needed, and where they acquired? In science. They are acquired in science. When you want to analyze data, when you want to conclude an observation, when you want to measure, have some measurements, get the length, when you want to handle the apparatus, when you want to see the features that a particular specimen or object or an item has, you have to observe. Number 2, the table below shows SI units. So SI units stand for International System of Units of some quantities. Remember, these are just units generated by a group of scientists who sat back then in 19th. So in 19th century, there were a group of scientists who sat down and realized that for these particular items, for these particular products, to have a common name within the universe, we have to give them what is called SI units. So there is where SI units originated from. Fill in the table by stating the quantity, and if the quantity is a basic quantity or derived quantity. Good. So let's go. Cubic meters. Now, when you talk about cubic meters, that quantity is volume. Volume is measured in cubic meters, and volume is a derived quantity. Volume is a derived quantity. You have to find the measurement, or you have to find the SI unit by calculating 2 or more. You must have 2 or more measurements so that you find the quantity. Number 2, ampere. So ampere. Now this one is for electric current. Electric current. Electric current, that is a basic quantity. Basic quantity. Number C, mole. Now, mole is the amount of substance. In that case, mole is the amount of substance. Amount of what? Of substance. Now, mole is a basic quantity. A basic quantity. Number 4, candela. So candela, that is light intensity. That is the quantity for it. light intensity. If you want to measure how strong, or rather how low the intensity of light is, intensity. And that one is, again, is a basic quantity. Let's move to kilogram per cubic meter. This one is for density. Density is measured in kilogram per cubic meter. That one is a derived quantity, where we must have the measurements for mass and the measurements for volume put together. So that one is a derived quantity. And then seconds, that is time. Time is measured in seconds. That one is a basic quantity. A basic quantity. So by just doing so, you already have 6 marks. Just providing the quantity. Cubic meters, that is the measurement for volume. Remember, for us to find volume, we must have one of the measurement in meters, another one in meters, and another one in meters. So when these are multiplied together, they bring us what? Cubic meters. So in other words, derived quantities must be calculated from 2 or more what? 2 or more measurements. But basic quantities, their units are already known. So they have already known units. When you go to amperes, that is electric current, we just find it direct. Amount of substance, the mole, again. Light intensity, and when we go to kilogram and seconds, those ones are both derived. Are both derived and what? Are either derived or basic quantity. Number 3, the diagram below shows a Bunsen burner. So this one again is a grade 7 work, Bunsen burner. The part labeled W, this one is the chimney, or the chimney can be called barrel. The part labeled K, this one is collar. Now let's move. Let's dive to the question. Identify the parts W. Just like I've mentioned, W is the chimney. And we are also going to accept barrel. I'm not saying that you as a learner to accept. This one is for the teachers. I'm just trying to tell the person who will get this paper again, including the teachers, that they accept the answer what? The answer called barrel. So barrel is also the chimney part. K is the collar. K is the collar. K is the collar. Let's move to 4. I hope we are together. And if you want to access Kenya Science, if you want to reach us out, the Kenya Science access number is 0790-199-736. Don't forget to subscribe, because this is one of the channels that all learners within Kenya, just like the name says, Kenya Science Academy, have joined. So if you are left out, remember you're losing a lot. So welcome to Kenya Science. Number 4. The diagram below shows a setup which was used during the integrated science lesson. So we have a setup here. There is a salt and water solution. Remember, when salt is added to water, the salt crystals are able to dissolve. And then when they are heated, just like we are seeing here, there's a Bunsen burner heating it. And this is called a tripod stand. So the round bottom flask is placed on the tripod stand and being heated. The solution is heated, and then you can see cork here. Cork is to prevent any entry of air. So it therefore means that whatever we are heating will just concentrate on the delivery tube and be collected at the end of the experiment here, which is called condensed water, because we have cold water. So this cool water, or rather cold water, is able to now cool the vapor that is being delivered through or via the delivery tube to the boiling tube. So that is the work of cold water in this case. It is able to cool the vapor as it condenses to what? Back to liquid. Identify the method of separating mixtures shown. So this method is called simple distillation. Simple distillation. So whatever that is being cooled here is called a distillate. Whatever that is being cooled here is called a distillate. Name two mixtures that can be separated using the setup. So number one mixture is already there, salt and water, sugar and water. Those are some of the mixtures that can be separated using the setup. So in this case, we are trying to separate those solids that are soluble in water or in liquid. So in this case, simple distillation is talking about solids that are soluble in liquids. So you look at water, it is a liquid. And then you look at salt and sugar, they are solids. So in that case, when we pour in sugar or salt in water, what is going to happen is that all of them are going to disappear in that water. So it therefore means, in other words, that it has dissolved. So those are some of the two examples of mixtures that can be separated using the setup above. And remember, as a learner, you already been given salt and water solution. That is a mixture. That is a mixture. Let's move to number 5. Name the equipment that will be used to perform the following terms in the laboratory. In the laboratory. Now, let's move on. The equipment that is used to perform the following in the laboratory. Number 1, holding solid substances when heating. So when you want to hold a solid substance when heating, number 1, we can use a beaker. Number 2, we can use a test tube. Number 3, we can use a boiling tube. Still, we are holding substances. Are you getting that? So those are some of the substances that we can use in holding solid. So when you put in solid substances in a beaker, they will be held. You put them in test tube, they will be held. In a boiling tube, they will be held. Transferring small amounts of fixed volume of liquid. So when you talk about that, we are talking about pipette in that case. Pipette will only be able to transfer the fixed amount of what? Of liquids. Now, in this case, dropper is not the best option in this case because we are talking about the fixed amount. But because we know that pipette is able to carry 25 ml of the liquid, then it therefore means that is a fixed what? That is a fixed amount. So let's move to number 6. Grade 7 learners were learning on human excretory system. We know that human excretory system, this is a system that it helps in eliminating waste products out of the body. Eliminating what? Waste products. Waste products. And one of these waste products include the sweat. Sweat is being excreted via the skin. And remember, sweat is composed of what? We have water, salt. Those are some of the components of what? Of sweat. Highlighted human skin as one of them. Yes. He later drew the human skin below. Identify the parts marked A to D. Now, the parts marked A, in this case, this is epidermis, which is the outermost layer of the skin. Part marked B, this is the dermis, the inner layer. And then part marked C is the fat layer. We call it fat layer, or it can be called subcutaneous layer. Subcutaneous layer. And the part marked D, this is the air. The air. Now, oh, that was the question. They're just identifying the parts. Amazing. So those are just the 4 marks. 4 marks for free. Just identifying the parts. Other parts include this area, which is sometimes common. This is called sweat duct. And down here is called sweat gland. And there are 3 functions of sweat gland. Number 1, sweat gland helps regulate body temperature. Number 2, sweat gland helps excrete wastes. Excrete wastes. And last one, the role of sweat gland is to produce sweat. That is the most common role of sweat gland. Produce what? Sweat. Other student who doesn't want to use the word produce will always use the word secrete. He will always use the word secrete sweat. All means the same. Yes, let's move to number 7. If you have any question, you will comment down there. Ask me, Teacher Zamalek, I didn't understand this. May you explain further. I'm here for you. That is why Kenya Science is always one of the best schools, online schools that learners will always come listen to, whatever that is being taught by most of the professionals or professional teachers. Now, integrated science, that is number 7, is a broad area of study. That is true. That involves the study of various branches of science together. Name the 3 main components of integrated science. So what do we study mostly in integrated science? Number 1, we study chemistry. That is one of the integral part. Another part is the biology. That is another thing that we study in chemistry, in integrated science, biology. And the last one, which we majorly study here, is physics. Physics. So these are the 3 branches or main components of integrated science or sub-disciplines of integrated science. If the question comes, integrated science has how many sub-disciplines? Talk about chemistry, biology, and physics. How many components? Talk about the 3. Unless if they ask you 5. If they ask you 5, number 4, we can talk about astronomy. And number 5, we talk about earth science. So in that case, you are allowed to add the 2. You are allowed to add the 2. Let's move to number 8. List 2 apparatus that are used for measuring accurate volumes of liquids. 2 marks. Wow. That one is very, very simple. They are used to measure accurate volumes of liquids. Number 1, we have pipette. Remember I talked about pipette that time? And pipette always measure a volume of 25 ml. Exact. Number 2 is burette. Again, burette measures up to 50 ml. Number 3, which is very common, which is a common one, is measuring cylinder. And in most cases, they usually ask this question. If it comes to paper 1, integrated science, measuring cylinder, section A. Remember, science has got two sections, section A and section B. And in most cases, that question of the apparatus that is used to measure accurate volumes of liquids, the answer is usually what? Measuring cylinders. Number 9, differentiate between pure and impure substances based on melting point and boiling point. Wow. This question is very, very common. A very, very common question. Now, let's move on. Pure substances. Pure substances have a fixed melting point. They have a fixed melting point and boiling point. They have a fixed melting point and what? Boiling point. Now, for example, water. Water is one of the substances with a pure melting point. Water has a melting point of 0 degrees Celsius. That is the melting point of water. And a boiling point . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . of 100 degrees what? Celsius. So in that case, it therefore means that pure substances, water is one of the pure substances we are mentioning in this case. So it therefore means that it has a fixed melting point and boiling point. If the question comes like it has come without asking you, requesting you to give examples, don't give like I have given. Just fix your answer to having a fixed melting point and boiling point. Stop by there. Now, while impure substances have a range of values of melting point, a range of values of temperatures. Let's talk about temperatures of the melting point and boiling point of substances. So it means that there is a range of values of temperatures. Now we don't know. When you look at the melting point of a candle is one of them, and the boiling point of a candle, it differs. So there is a range. Sometimes it's 64, sometimes it is 60, sometimes it is 68. So we are not certain that this is the exact melting point of a candle. This is the exact boiling point of a candle. So candle is one of the impure substances. Number 10. Now, the diagram below represents heating curve of pure and impure substances. Which curve represent a pure substance? Now, the one with the sharp arrow like this one is a sharp one. This one is a pure substance. But the one with a sigmoid, or rather an S form, that one is an impure substance. An impure. So just like I have said, fixed, sure, direct. That is a pure substance. But melting point, it ranges. You can easily see how this particular A of a substance behaves when heated. Identify two sexually transmitted diseases. Two sexually transmitted. Now, STDs. Number 1, HIV and AIDS. Number 2, we have syphilis. Number 3, we can talk about gonorrhea. So those are some of the sexually transmitted what? Diseases. Let's move to number 12. Say what the following signs mean. Say what the following signs mean. This one, of course, the answers are there. Fire assembly point. So this simply means fire assembly point. So we can assemble in case of fire outbreak. So this is where we converge. So this is where we do what? We converge. There is no space. So if you have any question about that, just comment. I will explain more. So this is fire exit or escape in case of fire outbreak. And then you come to fire alarm. This is for alert. So for alerting. So you have to alert us, eh? So this is escape. Number 13, name the following parts of a cell. Name the following parts of a cell. Now the first part here, this part is called a cell membrane. And this is an animal cell, eh? This is an animal cell as seen under a light microscope. So this is called cell membrane, or it can also be called plasma membrane. Plasma what? Plasma membrane. The other name is plasmalemma. Plasmalemma. That is the other name. The second part, this is the nucleus. Nucleus. And the last one is the cytoplasm. The last one is the cytoplasm. Cytoplasm. Let's move to number 14. Outline the functions of the following parts of a cell. Right, whether found in plants or animals or both. Amazing. Vacuole. Now, you look at this particular cell we have seen here. There is no vacuole. But when you come here, function of the vacuole is that it stores. So it stores. This stores nutrients. It stores sugar, stores water, stores salts, and other minerals. So this one is found in both. This one is found in both. If we move to cell membrane allows movement of substances, in and out of the cell. Allows movement of substances in and out of the cell. So both have a cell membrane. Cytoplasm. I mean, the vacuole is found in plant cells. Sorry for that. This one is found in plant cell. Now let's move to, sometimes at some point the vacuole may be small. It might appear to be very, very small. So in major cases, they are found in plant cell. Cytoplasm. Now, here is where it is a site where all chemical activities takes place. That is where all chemical activities does what? Takes place. So both have a cytoplasm. The only difference is that you look at the cytoplasm of an animal cell, it's all over the cell. But when you look at that of a plant cell, they are concentrated basically towards the periphery, just like the nucleus. So they also have nucleus, which controls all the activities of the cell. Controls all the activity of the what? Of the cell. Another function is that it stores DNA. The genetic information. The genetic what? Information. And that is why at some point you want to be dark, just like one of your parents. You can be chocolate in color, just like one of your parents. You can have the shape, the shape or the organization of the structures of your body, just like one of your parents. So it means that those are characteristics. Where do they be stored? They are stored in the nucleus. Nucleus have them, and they are stored specifically in the DNA. So that is where we have genetic information. Information about your parents are being passed through you during reproduction process. So nucleus is also present in both. I think that marks the end. That marks the end of that question. Let's move to number 15. Mwangi placed blue ink in a conical flask that had water in it. The water finally turned blue. Wow. Question. This process is called diffusion. So you can easily see that the particles are moving. Once they have been dropped into the liquid, they are now moving from the region of high concentration here to their region where they are lowly concentrated, and they are able to spread out through the liquid. That process is called diffusion. Number 16. Grade 8 learners conducted an experiment to as demonstrated below. So there is an experiment here. There is whisking tubing, water, and there is dried concentrated sugar solution. So you can see there is initial level of solution. So you can easily see. After some times, the final level of solution. So the solution increased in volume, dried concentrated sugar solution, and there is also water. Now, what happens at the level of dye? It should be dye. Anyway, dye, concentrated sugar solution in the capillary after 20 minutes. So the volume, the level of the dye. So the volume of the dyed concentrated solution, concentrated sugar solution, increased. So the volume did what? Increase. So you can easily see that initially the volume was around here. The initial volume is here. But after some times, the experiment, the level rose up to this point. So you can easily see that. What did the above change? Why did the above change take place? Now, the above change took place due to osmosis. Due to osmosis. What happened here? Now, you can easily see that the dyed concentrated sugar solution is within the whisking tubing or the capillary tube. And the whisking tubing is also inserted inside the capillary tube. So water from the beaker now entered through the whisking tubing onto the capillary tube to increase the volume at this moment. So therefore means that water moves from the region where they are highly concentrated, high water concentration, to region of low water concentration. So what happens is that this water now will reduce in size. So in volume. Initially, they have said that what happens to the level of that one in the capillary after 20 minutes. Oh, sorry. The volume of the dyed concentrated sugar solution increased. So you have seen that this particular dyed concentrated sugar solution is within the whisking tubing, within the capillary tube, but water is within the beaker. So what happens is that water entered and formed a solution with the sugar solution in the capillary tube, increasing the volume through a process called osmosis. So water moved from their region of where they are high to the region where they are low. And remember, where the water enters, where we have sugar solution, it is highly concentrated than where we have the water. Where we have water is highly water concentrated. I want you to understand that there is what we call water concentrated, and there is what we call sugar or high concentration. Remember, when there is high concentration, the solute are several or are so many. But where we have dilute or low concentration, the water is there. So remember, water dilutes the sugar. Now, we cannot talk about high concentration where we have water alone. Where we have water alone here is lowly concentrated than where we have the sugar solution. So get the difference. So what happens is that after 20 minutes, the volume of the dyed concentrated sugar solution increased. So you can see that it increased. And then we can add and say, and the volume of water in the beaker, in the beaker, decreased. Decreased. So those are some of the changes that happened after 20 minutes. The volume of the dyed concentrated sugar solution did what? Increased in volume. But after that, the volume of water did what? Decreased. Why did that happen? Due to osmosis. So we can say where, due to osmosis, where the water molecules moved from their region, of high water concentration. We must mention high water concentration. Without high water concentration, we are now not talking about osmosis. When you just talk about high concentration, sorry, that case is osmosis. That case is diffusion. Remember, diffusion is high concentration to low concentration, or a region that is highly concentrated to a region that is lowly concentrated. But because we're talking about water moving, osmosis, we are supposed to mention high water. There must be water there. Because where there is a lot of water, there is high water concentration. But when we want to talk about concentration alone, minus not mentioning water, we have to now begin from low to high. To region of high water concentration, to their region of low water concentration. Low water concentration. So you can easily see that the initial beaker had a lot of water. So that is high water concentration. But then it is lowly concentrated because of high sugar concentration. And then when you come to after 20 minutes, the volume of water in the beaker decreased because water had already moved through the whisking tubing or the semi-permeable membrane. That is the other name of whisking tubing, through the process called osmosis. So we can say, due to osmosis, where the water molecules move from the region of high water concentration to the region of low water concentration, increasing the volume, or we can explain later. That is under explanation. In fact, why did the above change takes place? Because of osmosis. That is just one mark. Because marks are not given here, we need to just predict. So we can say due to osmosis, where the water moved from the region of high water concentration to the region of low water concentration across a semi-permeable membrane. And in that case, it is the whisking tubing. Next question. Explain the biological process taking place in the experiment. That is just osmosis. Again, we are repeating what we have just written. Osmosis. That is the biological process in that case. The change took place because, again, we can just explain the change took place because or the first experiment, the capillary tube concentration of the sugar was high and the volume was low. You look at the second one, the concentration was low, the volume was high. So those all happens due to osmosis. Explain osmosis. So we are explaining the meaning of osmosis. Explain the biological process. So, here we have talked about moved. We are trying to explain why the change took place. Now we want to define, we are explaining what is osmosis. That is the biological process under the experiment. Osmosis now, it is the movement. Let me talk about net movement. It is the net movement of water molecules from their region from their region of high water concentration here is where learners get it wrong. High water concentration to their region of low water concentration across a semi-permeable membrane. Semi-permeable membrane. So that is what the particular term means. The last question under the experiment. What is the role of whisking tubing? What is the role of whisking tubing? What is the role of whisking tubing in the experiment? The role of whisking tubing in the experiment. The role of whisking tubing in the experiment. Now, we know that the whisking tubing, its role, it acts as a semi-permeable membrane. Semi-permeable membrane. It acts as a semi-permeable membrane, allowing only smaller sized molecules to pass through it as it blocks other larger molecules. So that is the role of whisking tubing. If it is 1 mark, semi-permeable membrane. If it is 2 marks, or rather explain, it allows smaller molecules to pass through it as it blocks what? The larger one. What can we compare the whisking tubing with in a living cell? 4 marks. No, this should be 1 mark. This should be 1 mark. So that is just the semi-permeable membrane or cell membrane. In fact, we can compare it with cell membrane. So our answer in that case is what? Cell membrane or plasma membrane. Remember, they're talking with a living cell. Plasma what? Membrane. Because it is semi-permeable. Or plasma lemma. Good. Number 18. Number 18, there is a diagram, but then this diagram is not supposed to be here. So we are going to skip that one. Number 18, define the following terms. Remember, this one is a diagram for a simple circuit. I don't know whether this diagram should be down here or not. I'm going to see that one later on. Let's dive to question 18. Define the following terms. Atom. Now, atom is the smallest particle of an element. The smallest particle of an element that can take part in a chemical reaction. That can take part in a chemical reaction. Yes, that is the meaning of an atom. It is the smallest particle. Remember, particles are made up of solid, liquid, and gases, those particles. But now the smallest one. And it can take part in a reaction. That is what we call an atom. Let's move to an element. Now, an element is a pure substance. A pure substance. That cannot be broken. That cannot be broken. Cannot be broken down into simpler substances. Into simpler substances. by either chemical or physical means. By either chemical or physical means. So that is what an element is. Now, for instance, you look at the 20 elements of the periodic table. Number 1 is hydrogen, up to number 20, which is calcium. Remember, we have almost 119 elements existing on Earth, the ones that have been discovered. But there are others that scientists are continuously discovering right from the day to late in the night. So these elements, they are essential on Earth. And that is why we have to discover them. We have to know them. We have to recognize them by their functions, their names, how they react. For instance, oxygen. How is oxygen reacting? How is oxygen helpful? So how can we harness oxygen and use that oxygen? So those are elements. They are elements. And remember, they are pure. And that is why you use them in the living cells. One of them is oxygen. They cannot be broken down. They cannot be broken down. Let's move to molecules. So molecules are pure substances, again, are pure substances made up of two or more atoms, chemically combined. So that is what the meaning of a molecule is. Now, for instance, when we have one atom of oxygen reacted with another atom of oxygen, they form the oxygen molecule with how many? 2. This is 1 plus 1 equal to 2. So that is why we have molecules made, substances made up of 1 or 2 atoms. So oxygen atom, this is an atom. This is also an atom. They react chemically. That is what I'm saying about chemically combined. Let's move to the meaning of a compound. These 4 marks are simple to garner. Very simple. Number 4. What are compounds? Compounds refers to 2 or more substances, or elements, 2 or more elements chemically combined together. So, for example, when I have hydrogen plus oxygen, this is an element. This is also an element. I want you to get the difference between a molecule and a compound. When we talk about a molecule, we are talking about those of the same species. Now, hydrogen and a hydrogen atom, they give us a molecule of hydrogen, 2 molecules of hydrogen. So, when we talk about elements combining, it therefore means that number 1 element combining with number 8 element in the periodic table, they give us a compound. Are we together? When carbon, which is number 6 in the periodic table, combined with oxygen, which is number 8 in the periodic table, they form carbon 4 oxide, which is a molecule, a compound. When sodium with a symbol Na, number 11 in the periodic table, react with chlorine, which is number 17 in the periodic table, a compound is formed called sodium chloride. This is a compound. So when you talk about compound, it is the elements combining. When you talk about molecule, it is the atoms combining. And how are they combining? They combine chemically. They combine chemically. Number 19. If you have a question, always contact Kenya Science. The number is always 07 90 199 736. That is the access number to Kenya Science. Let's move to number 19. A patient arrived in the hospital with swollen legs and was diagnosed with kidney disorders. List 2 causes of kidney disorders. what can cause a kidney disorder? Number one is diabetes. Now, if you have either diabetes mellitus or you have the other type of diabetes, you are going to have a kidney disorder. Remember, the kidney always filter blood to remove wastes. So one of the waste that is being removed by the kidney is the urine, which gets out of the body via the urethra. So what happens is that once the kidney has do what is called filtration, the urine is formed, and that urine is formed from urea, excess water, excess salt, and other substances which might be also present, like amino acid, in the urine. So that makes all collectively a urine. So urine, now once it is made, it is being transported, sorry for that, it is being transported from the kidney to the bladder via what we call ureter. Ureter, these are just tubes. There are two, one on the left side and one on the right side. So once the urine has been formed, these particular waste products are supposed to be eliminated out of the body. Why is that so? Because once the waste products accumulates in the body, they will have killed our cells. And remember, when the cells are killed, you can die. So we need to always eliminate these particular waste products out of the body. Now, for instance, a lot of urine accumulates in your bladder. They are not eliminated out of the body. They are not excreted out of the body. The kidney or the excretory system will have disorders. And that disorder is what, one of them is the kidney disorders. So diabetes is one of them. Another one is untreated infections. Untreated infections. Always treat the infections, particularly the UTIs. Kindly make sure that they are being treated. Another one is excess consumption of alcohol. Excess consumption of alcohol. So that one can cause kidney disorders. Remember, when the alcohol is too much for the kidney to do what is called detoxify. So just to reduce or completely eliminate the toxicity of the alcohol as a substance in your body, then it is going to malfunction. If it experiences too much work for it, it will now undergo a problem. And that problem or challenge will result into kidney disorder. So excess consumption of alcohol or just consumption, too much of it, will cause kidney disorders. Another one is high blood pressure. High blood pressure. High blood pressure. So again, kidney disorders are brought by when the blood is moving with a high pressure. Number 20, let's go. Write the chemical symbols of each of the following elements listed below. The chemical symbols of each of the elements listed below. This is so simple. 4 marks. So simple. Helium. The symbol for helium is He. Remember, there's a difference between helium symbol and hydrogen symbol. That for hydrogen is just H alone. But for symbol, there must be capital H and small e. And then fluorine. Fluorine is capital F. Fluorine is capital F. Remember, fluorine is found where? In the toothpaste. So that is one of the commercial function of the element fluorine. These all elements are very, very important. They are very, very important. Like when you talk about this helium in the balloon, it is one of the fuel also. So look at how it is very important. When you come to aluminum, the symbol for aluminum is capital A, small l. Sodium is capital N, small a. Good. So you garner all the 4 marks. Let's move to number 21. Number 21, you are told to complete the table. And I want you to do me number 21 and send me your answers via the comment section. You can send the answers via the comment section, or you can always call us or send the answers via the number 0790199736. As a learner, kindly, I'm requesting you, because we need to learn, and learning entails me and you. That is when I'll know that we have learned something. So I want you to go and do me number 21. Please, you who is going through this paper, do number 21 for me. Color in phenolphthalein. What is the color of wood ash? Remember, wood ash is always basic. This is a basic solution, or in other words, it is called an alkaline solution. When you come to lemon juice is acidic. It is acidic. It is acidic. Anti-acid tablet solution, this one is basic again. When you come to toothpaste, this one is also basic. So I would want you to give me the color in phenolphthalein, color in litmus paper, color in methyl orange. These are indicators. They are called commercial indicators. That is, again, plant extract indicators. So, for instance, when I talk about color of wood ash in litmus paper, wood ash is basic. So, when you bring the color in litmus paper, base will turn, when it is red litmus paper, so red turn to blue, and blue remain blue. So you are going to give me the colors. You're going to give me the color in litmus paper. When you come to, let's talk about lemon juice, acidic. So the blue litmus paper turn to red, and the red litmus paper remain red. So I expect you to answer such like question in the same manner. Are you getting that? So you just say blue, red, and so forth and so forth. Number 22. Remember to do me number 21, please. You can send your answers via the number, or you can type down within the comment section your answers. Thank you for that. Thank you for accepting that. Identify the elements that make up the following compounds. Number 22, sodium bromide. So sodium bromide is composed of sodium and bromine. Sodium and bromine. Magnesium nitrate. In symbol, magnesium nitrate is written this way. Magnesium Mg. Nitrate is NO3. And then there is 2 here. There is 2 there. So let's go. Let's move on. Magnesium nitrate. It is composed of magnesium as an element. It is composed of nitrogen and oxygen. And then magnesium nitrate. So this one is just a repetition for this one. That one is the repetition for the other one. So those are the possible what? The possible answers. Look at the picture of the electric circuit below. Name the parts labeled P. So P, this is the switch. Not the switch, but the battery or the dry cell. So you can see the positive and the negative. So this is the dry cell or battery. We don't say battery, but we say battery or battery. Yeah, Q. Now Q is the switch. Q now is the switch. R. R is the bulb. The light bulb. R is the light what? The light bulb. That was a simple electric what? Circuit. As simple as that. As simple as that. Now, let's move on. Identify the elements and compounds that are used in making the following. Toothpaste. I talked about fluorine or fluoride. And we can talk about calcium carbonate. But let's only talk about the chlorine. The fluorine. And then when we move to soap and detergents, so we are going to talk about sodium salts or sodium components. So let me talk about sodium hydroxide. That one is the only compound that is present in soaps and detergents. Sodium what? Hydroxide. Remember the question was specific, elements and compounds. And one of the elements in toothpaste, we have talked about fluorine. Table salt. Table salt. Again, table salt is sodium chloride. The compound that is present in table salt, the salt that you use at home in various food to add flavor. That salt is called sodium chloride. Go and look at the polythene paper, that is holding or housing the salt. You are going to find out it is written sodium chloride. Sodium chloride. Now, I'm going to come back in number 20 from number 25. Let's end our lesson from number 24 for today. And the rest of the questions I will do tomorrow, or rather next time. So thank you for your time. Kindly go through the paper. I will come back with section A and the last section of this paper. It was a very, very good paper. You can look at it. A very, very nice one. For those who are in grade 9, this one is a very good paper for you. Kindly. It entails grade 7, grade 8, and grade 9 work. Go through the paper. The paper is meant for you. This paper, once you revise it, you will be good to go. Remember, all the topics are here. All the topics are here. And because the paper is too long, that is why I want to end it from there. Too much of something is dangerous. Too much of something is dangerous. We only have to just state or mention or reach our limit capacity. And that is why I will come back with question 25, question 26, question 27, question 28. You can see 29, 30. The questions are still more. So I want to end my session from there. I want to request you to do question 21 for me, and send your responses within the comment section down there. Too much of something is dangerous. Thank you for your time. See you next time. Your teacher is gone.